Okay, so here we are on lecture 2. We're going to talk about the S phase and DNA replication in this one. So again, this is probably something that you guys have seen before, and I doubt there's a whole lot of detail in here that you haven't seen. In fact, we're not going to go through a whole lot of the details. That'll be more for a genetics class than a cell class. So let's just work our way through it pretty quickly. There will be a couple of new things in here, I think, for you guys, but the reason why we're putting them in here is because when it comes to the regulation phase, they'll be important for something we'll discuss later. So S phase, the S actually stands for synthesis, and of course, what you guys are familiar with in DNA replication and the synthesis of those DNA copies of the chromosomes is the primary event that's going to take place, but there's actually a lot of stuff the cell is going to make during this time. So remember last time we talked about a restriction point, a point at which cells, as they are doing what they do during G1, they decide to divide, and when it comes time to divide, there's certain things that have to happen in order for them to do so. So one of the things that has to happen over the course of the S phase and in G2 is that the cell's going to have to increase in size. So cells are going to actually increase in size before they divide. So we can't take an existing cell and divide it in two. Those daughter cells will be too small and not actually get all the components they need. So for division, we've got to increase our size, we've got to make new organelles, we've got to make a copy of our DNA, we've got to split all of our other structures, we've got to make sure we have enough ribosomes. All of these kinds of things have to take place during S phase. So definitely think bigger than just DNA replication, but of course DNA replication will be a pretty important process, and that's why we're going to talk about it a little bit more in detail. And one of the main reasons is because DNA replication is one of the main controllers of how we work our way through this process. Remember that DNA replication is semi-conservative. What that means is you start with that strand of DNA, you pull it apart, and this side's going to get a duplicate, this side's going to get a duplicate. So each one of the new strands in is going to have an original and then a brand new copy. And remember, this will be based on the base pairs. So if there's an A, we put a T. If there's a G, we put a C, and so on, as we work our way across this process. And so this would be what semi-conservative means. The replication process is going to begin at specific regions called origins of replication. These guys tend to be rich in AT base pairs. Hopefully that makes sense to you guys if you think about that first process where we have to peel the two DNA strands apart so we can make copies of both sides. It hopefully will make sense that we would pull the helix apart at an AT rich area, largely because of remember, AT bonds or AT base pairs have 2 hydrogen bonds instead of 3. So peeling apart an area like this would cost us less energy than a GC area. And one of the most important control aspects of one, how a cell is going to go through its replication phase, and two, one of the initial regulators of going through the cell cycle is going to be this mechanism by which replication is allowed to initiate. This is an important piece because the problems that could arise if we duplicated our DNA more than once, so we end up with multiple copies, can obviously cause an issue for those daughter cells. So if we are actually going to begin at multiple areas across the chromosomes, then it makes sense that we would have to have some mechanism in place to make sure that we only do each one of them once. So the mechanism that is doing this, and again, if you haven't had a genetics class, you may not have heard of these before. They're not one of the standard enzymes that we talk about in replication at an intro level, but these are what are called licensing factors. And basically what licensing factors do is that they are going to bind to an origin, and when they bind to the origin, that origin will be able to initiate replication. When they initiate replication, licensing factor gets detached and deactivated, so that origin basically becomes unlicensed and it will not promote replication a second time. So these guys are going to be downstream factors when it comes to cell division. So some of the growth factors and so on that we are going to talk about later are actually going to work their effects on a cell through these licensing factors. So that's why I want you to add these to your knowledge of replication at this point. Here's a pretty decent overview, if you will, of what replication looks like. You've got a few of the enzymes in there. So these, of course, would be the helicases. You guys are familiar with those as the unwinders. So these would be your origin and licensing factors. So CDT1 is going to be a big one, and these CDCs are going to be regulators. So we'll introduce those a little bit later. But these guys will be regulators. These guys are targets, and you can see they work together to initiate DNA replication. Here is, I think, a pretty good summary of the replication process. Again, I'm not going to, in this course, worry too much about all these enzymes, but you've heard of primase, DNA polymerase, ligase, Okazaki fragments, all that kind of stuff before. Definitely refresh your memory on that. This whole mechanism here, of course, is a replication fork, and you would have a second one on the other side where this would be a replication bubble. So let's see. I do want you to know that DNA polymerases, just because of the primary role that they play in synthesizing the DNA strand, and those bubbles are going to get bigger and bigger, and eventually they will combine with each other until eventually all of your chromosomes, so for us 46 chromosomes with multiple start points on them, are going to be completed. And it's really kind of remarkable because for us, those 46 chromosomes have about 6.4 billion nucleotides in them, and so that would be the number of bases that we have to put in place and or replicate during, or add during replication. And it's really quite remarkable that we basically can complete the process in about 9 hours ourselves. So here's another way of looking at it. So you'd have multiple origins, and these guys, these bubbles expand, and all these don't have to start at the same time. These bubbles could be different sizes. You can see that here. So here's one that's been going for a while. Here's one that just started. Here's one that's about somewhere in between. Another thing you can see here would be the packaging of the DNA. Remember those nucleosomes where the chromosomes are wrapped around those little proteins to create chromatin? That is found in front of the replication and is found behind the replication. So basically the packaging's adjusting as the replication takes place. Bottom line is, once we're done, we end up with 2 daughter molecules here of DNA. So this would be a double helix, this would be a double helix, and in the case of the chromosome, these are what are called sister chromatids. So eventually, I mean, we'll clarify this as we go along, but you definitely need to know the difference between a sister chromatid and homologous chromosomes. Those are not the same term. Sister chromatids are actually going to remain attached to each other by the centromere of the chromosome. So the centromere has not been duplicated yet. They will also be linked against each other with what are called cohesin proteins. So let's just look at a picture here. So here's one sister chromatid, another sister chromatid. This, of course, is during M phase, and everything's packed down tight, so it stains real dark. Cohesins would be found in here, where you would attach these to each other, and we'll talk about those later. And condensins are going to use these cohesins to actually create for us, so in prophase, when it's time to condense these down, those compact chromosomes like we just saw in that picture is actually the combination of cohesins and condensins. So condensins will come in M phase, and cohesins are added after the replication. Notice we're doing that in G2. Alright, so next we will dig into the M phase a little bit, and so just to prep you there, cell division actually is going to involve 2 major functions during M phase. And the first is going to be karyokinesis. This is the division of the nucleus. And this is where mitosis and meiosis are going to do their jobs. And then about the same time, it actually starts a little bit later, about halfway through, the division of the cell is going to start, and this is what we call cytokinesis. So karyokinesis and cytokinesis are two separate events, although very well coordinated. And as we'll see, this one's going to involve the nucleus. We'll also see the microtubules involved here, and then cytokinesis is going to involve the microfilaments. Remember, the cell's going to basically contract and split in two. Okay? All right, so next time what we'll do is we'll move through the phases of M phase and look at some of the main components of those. Bye.